Hey, I'm Dr. Steve Mailer, and we are talking about a really exciting technology today. We're going to go over plant-made biologics versus CHO cells, and is the future of medicine growing in a greenhouse? Think about it. Every time you receive a monoclonal antibody, whether it's for cancer, autoimmune disease, inflammatory bowel disease, or even a vaccine, there's a good chance it was made inside millions of living hamster cells. Yes, hamster cells, specifically the Chinese hamster ovary cells, which are known as CHO cells. For nearly 40 years, they've been the gold standard for manufacturing biological drugs. But a new challenger has emerged: plants. So, could greenhouses someday replace billion-dollar biomanufacturing facilities, or is this just another biotech hype cycle? Today, we're comparing the current king of biologic manufacturing, those CHO cells, with one of the most exciting emerging technology in pharmaceutical science: plant-made biologics. So, why are biologics so hard to make? Unlike aspirin or penicillin, biologics aren't chemicals, they're proteins, extremely large, three-dimensional, intricately folded proteins. A monoclonal antibody contains over 1,300 amino acids arranged into an incredibly precise structure. If that protein folds incorrectly anywhere, it may not work. If the wrong sugars are attached, if the immune system may recognize it differently, if impurities remain, the patients can become very sick. So making biologics isn't chemistry, it's cell biology. And why CHO cells became the gold standard was back in the 1980s, scientists faced a problem. Human proteins require complex cellular machinery. Bacteria couldn't perform many of the post-translational modifications that are needed for therapeutic proteins. And CHO cells solve that problem beautifully. These mammalian cells naturally perform many of the same processes as human cells do. They correctly fold proteins and form disulfide bonds and assemble antibodies, and they attach complex sugars and they secrete proteins into cultured media. So today, more than 70% of approved recombinant therapeutic proteins are produced in CHO cells. Virtually every blockbuster monoclonal antibody you've heard of relies on this technology. CHO manufacturing works, and regulators know how it works. The experience is incredibly valuable. But CHO manufacturing has problems, 40 years of problems. Success doesn't mean perfection, and producing biologics in mammalian cells is expensive. So imagine maintaining billions of living cells every second for weeks. They need special nutrients and strict temperature control and controlled oxygen and precise pH and sterile conditions and continuous monitoring. One bacterial contamination, one fungal spore can destroy an entire production batch worth millions of dollars. So building one manufacturing facility may cost hundreds of millions or even billions of dollars, and these costs eventually affect every patient receiving the medication. Enter plants. Now imagine replacing giant stainless steel bioreactors with tobacco plants, not because patients will smoke medicine, but because one species has become one of the biotechnology's favorite protein factories. So instead of growing hamster cells, scientists grow plants. Then they temporarily introduce DNA encoding the desired therapeutic protein, often using engineered viral vectors or a specific bacterium. Within days, the plant's own cells begin manufacturing that protein. The leaves are harvested, the protein's extracted, purified, tested, and formulated exactly like biologics made using any other manufacturing platform. The patient never receives plant tissue, only purified medicine. So, sounds pretty good. Which system is better? The answer depends on what you're trying to manufacture. So let's compare them. Let's look at speed. CHO cells often require months to establish stable production lines. Plants can frequently begin producing proteins within days after transient gene expression. That makes plant systems particularly attractive during outbreaks or when scientists need to rapidly screen therapeutic candidates. The winner here with speed? Plants. Now let's look at manufacturing cost. Plants don't require enormous sterile fermenters. They don't need expensive cell culture media. Scaling production may simply mean growing more plants. So several companies developing plant molecular farmings estimate dramatically lower upstream production costs, although the final cost of purified medicine still depends on downstream purification, quality testing, and the regulatory requirements. But the likely advantage here with cost? Plants. Let's talk about protein quality, and here's where things really become interesting. CHO cells evolved as mammals. Plants didn't. And that matters because proteins aren't finished when they're made. Cells decorate proteins with complex sugar molecules in a process called glycosylation, and those sugars will influence stability and half-life and immune recognition and biological activity. And historically, plant sugars are different than human sugars. And that raised concerns about whether plant-produced antibodies could function identically. But today, scientists can genetically engineer plants to produce much more human-like glycosylation patterns. The field is called glycoengineering. It's one of the biggest reasons plant-made biologics have advanced so rapidly over the last decade. So CHO cells still have the advantage for many highly complex proteins, but that gap continues to narrow. Now let's look at the regulatory experience, and this may actually be CHO's biggest advantage. Regulators know how CHO manufacturing works, and they know it extremely well over the last 40, 50 years. Thousands of production lots, hundreds of approved biologics, decades of safety data, and plant manufacturing simply doesn't have that same history yet. That doesn't mean it's inferior. It means it has more evidence to generate before becoming equally established. And AI changes everything. Here's something really fascinating. Artificial intelligence is becoming extraordinarily good at designing new antibodies. The bottleneck isn't always discovery anymore. It's manufacturing enough candidates to test. So imagine AI generating 5,000 possible antibodies, where traditional manufacturing can't realistically produce all of them quickly. Plant systems may allow researchers to manufacture and screen many more candidates in a fraction of the time. Now that combination, AI plus rapid plant production, fundamentally changes early drug discovery. Now, veterinary medicine may benefit even more. I'm a veterinarian, I'm a boarded surgeon, and this is where I particularly get excited because many biologics never make it into veterinary medicine, not because they don't work, because they cost way too much to manufacture. So imagine affordable monoclonal antibodies for immune-mediated disease, for targeted cancer therapies, for species-specific cytokines, novel vaccines, regenerative proteins. Lower manufacturing costs could completely change what's economically possible in veterinary medicine. So the big question is, will plants replace CHO cells? Probably not, and probably not anytime soon, at least not completely. Think about airplanes, helicopters, drones. They each fly, but each serves a very different purpose. So the future probably isn't one manufacturing platform replacing another. It's choosing the best system for each specific biologic. So some drugs will remain produced in mammalian cells and others may move to yeast and some to bacteria, and increasingly some may come from plants. So my final thoughts on this is the biggest breakthroughs in medicine don't always come from discovering new drugs. Sometimes they come from discovering better ways to manufacture them. CHO cells, they revolutionized biologics, but plant molecular farming may represent the next revolution, not because it replaces everything we've built, but because it expands on what's possible. Lower costs, faster development, greater manufacturing flexibility, improved global access, and perhaps most importantly, the ability to bring advanced biologic therapies to millions of patients, humans and animals alike. The future of medicine may not just be designed by artificial intelligence. It may also be grown in a greenhouse, and that's extraordinary thought. Thanks for listening. If you enjoy deep dives into biotechnology, veterinary medicine, and the science shaping the future of healthcare, be sure to subscribe. In our next episode, we're going to explore one of the biggest challenges in plant-made biologics, glycoengineering, and why a tiny sugar molecule can determine whether a life-saving drug succeeds or fails. Thanks for following me.